টুলটো হয়। Hello. 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 Yeah. Shall we start? Live people too. That means NPTEL and me only. Yeah, shall I start giving some lecture? Are there any attendees? Not yet. Hello. Yeah, shall I start speaking something or what shall I do? I can speak, right? Yeah. I do not see any participant. Are there any participant? No. Not yet. But this will be recorded. The student can see it later, right? Right. Anyway, okay, let me finish my job. Yeah, thank you. Welcome to my course on genome editing and engineering. Today we are having the second interactive session. So we try to cover 4 weeks in one open session or interactive session. And we also try to answer some of the queries put up by the students from time to time. Unfortunately, or maybe students have understood everything and there is no any query from their part. Anyway, I would like to sum up some of the things very briefly that are supposed to have been covered in these last 4-week courses. So mostly, as you can see, from week 5, we have discussions on zinc finger nucleases, which are very, very important for genome editing. So basically, there are lectures on the basics of zinc finger nucleases here. So there we describe how zinc finger nucleases were developed. So these are basically artificial nucleases, man-made nucleases, where components from two different proteins has been put up together. And then we go into discussing the design of the zinc finger nucleases for genome editing to make them much more efficient and target specific. So those details are already there. You have all gone through those lectures. I will try to sum up some of the important concepts regarding ZFN. And then finally, we go for the applications of zinc finger nucleases in the last lecture of this module. Now, you have those who have attended this course know about the zinc finger nucleases, which are actually basically groundbreaking molecular tools used in genetic engineering and gene therapy. And as I have already told you, and also you find in the lecture, they are designed to precisely modify the DNA with a specific target gene. These ZFNs are part of a broader class of genome editing technologies, along with other systems like CRISPR-Cas9 and TALENs. TALENs basically are transcription activity like effector nucleases. The key features of the zinc finger nucleases is their ability to create DSBs or double strand breaks. And in our earlier lectures, we have told you about the importance of DSBs, particularly from the gene editing or genome engineering point of view. So these ZFNs, which are basically engineered proteins, creates DSBs at precise locations within the genome. And these DSBs stimulate the cell's natural DNA repair machinery, which can lead to the introduction of desired genetic changes in the form of insertions, deletions, and substitutions, which are basically natural processes. So by this approach, we are basically trying to harness the cell's natural processes of DSB repair and insertion, deletion, and substitution mutations. In this design of, or in this scheme of things, the targeted DNA editing allows us to modify genes with great accuracy, potentially correcting genetic mutations responsible for various diseases, or creating genetically modified organisms for research and industrial applications. As I have already told you briefly, the structure of ZFN consists of two essential components. The first one is the zinc finger proteins, and a second was the nucleus domain. The zinc finger proteins serve as the targeting mechanism, and these proteins have a specific affinity for binding to specific sequences of DNA, effectively guiding the ZFN to the desired domain genomic location. zinc finger typically would recognize a 3 base pair segment of the DNA, as you already know, and by linking multiple zinc fingers together, we create a highly specific DNA binding domain. The nucleus domain of zinc finger nucleases is responsible for inducing the double strand breaks in the DNA at the specified location. It cleaves DNA strands, initiating the repair process, which we have just told you a little while ago. When the cell undergoes such a break, its repair mechanism is activated, and the cell tries to repair the double strand break, and these results in certain genetic modifications. These repair process occur through either homologous, non-homologous enjoining, which we call as NHEZ, which frequently introduces small insertions or deletions, or homology-directed repair or HDR mechanism, which allows for more precise gene editing. If you look into the various applications of this technology, gelatin has been used in various applications, or has the potential to be used in various applications like gene therapy, and then functional genomics, where researchers use these technology to study the function of specific genes by creating knockouts or introducing point mutations. And then specific domain applications in the area of, say, for example, agricultural biotechnology or biopharmaceutical production also has huge potential. For example, ZFNs have been employed to develop genetically modified crops or gene-edited crops, having improved traits such as resistance to pest or tolerance to environmental stress. In the biopharmaceutical sector, this technology can be used to enhance the production of therapeutic proteins in cell lines, improving the efficiency of the bioprocesses. While zinc finger nucleases have shown significant promise, they also have limitations in, for example, in the case of designing and engineering ZFNs, we may face a lot of difficulties because it's very complex and time consuming, and their specificity may not always be absolute, leading to off-target effects. So while understanding the power of technology, we also need to look to the flip side, and this is one of the disadvantages of this kind of technology. However, in recent years, newer genome editing technologies like CRISPR-Cas9 have evolved, and they are gaining popularity due to their simplicity and versatility, though they are also not free from, completely free from disadvantages. To sum up, ZFNs are a powerful tool for targeted genome editing, and it offers a precise control over genetic modifications. Their applications range from gene therapy to agriculture and biopharmaceutical production, and they are very important in the field of genetic engineering or genome editing. They have faced a competition of late from straightforward and accessible genome editing technologies in recent years, like CRISPR-Cas9. So this is the main, some of the. I'm trying to summarize some of the topics that we learned in week 5. Then there are other topics. For example, in week 7, we learn about the CRISPR-Cas9 technology, and how this system found in bacteria has now become a very important tool. So let me discuss about the CRISPR-Cas9 gene editing. Before that, let me try to see if there are some participants. There are no any participants, right? Am I correct? is live going on. Am I, John? Am I communication is on, right? I'm there. I need to verify once before. So can I get some help from input from NPTEL CET? Am I audible? Am I in communication mode? Okay, thank you. Yeah, thank you. Okay, I'm taking a small break. Let me see if somebody joins. I will sum up the lectures. Yeah. Okay, let me start. We have also dealt with the CRISPR-Cas9 technology in this in one of the modules, and as I already have mentioned prior to starting this discussion, that this was basically a system found in bacteria. Now it is one of the most important cutting edge tools, and which is revolutionizing the research in biology, as well as it is having potential applications in the area of therapeutics and many other fields like biofuel applications, developing climate resilient crops, so on and so forth. CRISPR-Cas9 gene editing is a revolutionary and highly versatile technology, and it has transformed the field of genetics and molecular biology. This is a short form for clustered, regularly interspaced short palindromic repeats, which are basically sequences found in the genomes of bacteria and archaea. And Cas9 refers to the protein used in this system. So in this module, we have discussed about the basics of CRISPR-Cas9, as well as also the applications of CRISPR-Cas9. So if we look into the lectures, lecture 20 was discussing about the CRISPR system in the bacteria. In two lectures, 20 and 21, and then we moved on to discussion about CRISPR-Cas9 in genome editing in another two lectures. Then finally, we ended up discussing the various applications of CRISPR-Cas9, and then also certain computational resources which are available for CRISPR-Cas system. Now, I want to highlight some of the important things about CRISPR-Cas9 genome editing tool. These CRISPR-Cas9 originally evolved as a defense mechanism in bacteria and archaea to protect against viral infections. Bacteria capture snippets of DNA or small pieces of DNA from viruses that have attacked them and incorporate these small pieces of DNA into their own genome within the CRISPR region, and this forms the basis of the memory of these attacks. And when there is a subsequent attack, this stored information is used to neutralize the virus. So another important concept is the gRNA or the guide RNA. So basically, these are designed synthetic pieces of RNA, which specifically, target particular DNA sequences of the interest. So these are created by scientists in the laboratory. These guide RNA acts as a molecular address during the Cas9 protein to the precise location in the genome that needs to be modified. Cas9 protein is an endonuclease enzyme that functions as molecular scissors. It binds to the gRNA and forms a complex. This complex scans the DNA in the cell, and when it encounters a DNA sequence that matches the gRNA, it makes a cut or a double-strand break at the location. And we have already discussed while summing up gene finger nucleases, the vital role of DSBs, how DSBs elicit the repair mechanism inside the cell, and due to which mutations like insertions, deletions, substitutions occur, leading to some genomic changes. So we also know that DNA repair is done by non-homologous enjoining and homologous directed repair. So this is similar in the case of all kinds of genome editing technologies which creates double strand breaks. And moving over to the applications of CRISPR-Cas9 gene editing, it's a very versatile platform or technology. Majority of the research work today is being carried out using these particular technology due to its simplicity. So in the lectures, we have given details of various application. Here, I would just like to give some summary. In the case of genetic engineering and gene editing, one of the main priorities has been in treating genetic diseases, and CRISPR-Cas9 holds a big promise for treating such genetic disorders by correcting disease-causing mutations at the DNA level. This technology has the capability to do genome editing at a single base level. So certain diseases which are being caused by single base change, or maybe SNPs, can be very easily cured by using these CRISPR-Cas9 technology. Now again, when it comes to agriculture and food security, which is very important for our globe, this technology has been used to modify the crops with desirable traits, just like your increasing the yield, pest resistance, or enhanced nutritional content. And in a climate change era, people are also trying to create climate resilient crops, and even crops which require less water to grow and produce food. In case of therapeutic development, newer therapies are being tried in the case of, for example, cancer immunotherapies by modifying immune cells to target cancer cells. And when it comes to species conservation or biodiversity, CRISPR-Cas9 has been explored for applications in conserving endangered species and preventing the spread of invasive species. So we know that CRISPR-Cas9 offers numerous benefits, and it has huge potential. But at the same time, it also poses ethical and safety concerns. And these are some of the issues we have highlighted from time to time. While discussing JDFN, we told that CRISPR-Cas9 is a better technology. It is having more efficiency. It is also simple. And JDFN has problems like off-target effects. CRISPR-Cas9, although it is better than JDFN, while other technology is not free from such problems. So we have, in this case, off-target effects too. Unintended genetic changes happens, and then ethical considerations surrounding its use in humans and environmental subjects of ongoing debate and regulation the world over. However, we need to understand that in spite of all these challenges, CRISPR-Cas9 has revolutionized gene editing, making it faster, cheaper, and more accessible than previous methods. And scientists are continuously refining and expanding its capabilities, and these offers huge potential to address a wide range of genetic diseases and advanced scientific understanding of the genome. So these are some of the things in brief we have learned in this module number week 7. So there is another technology which is a little bit older than CRISPR-Cas9, which is TALEN, and we have also tried to discuss the possibilities of TALEN and plant. So we started with a discussion on the basics of TALEN, and then design of TALEN for genome editing, and then we went for the applications of TALEN. In a similar way, we discussed about the CRISPR-Cas9. In the realm of genome editing, many important tools has been attempted, and some of them has been very, very popular. And amongst them, TALEN is a system which basically stands for transcription activator like effector nucleases, has emerged as a remarkable gene editing platform, and it is still considered as a rival to CRISPR-Cas9 in terms of precision and versatility. So if you look into the three technologies, ZFN, TALEN, and CRISPR-Cas9, and the order of their development, ZFN seems to have become a little bit less preferred technology. We cannot tell that it has lost the race. CRISPR-Cas9 is definitely leading the race, but TALEN is not less. It is a rival, or it is actually rivaling CRISPR-Cas9, as I already told you, in terms of precision and versatility. So we have learned about the TALEN system in brief in these lectures, and TALENs are derived from naturally occurring transcription activator like effectors or tails, which are proteins produced by certain plant pathogenic bacteria. These proteins have a unique ability to bind to specific DNA sequences in the host organism. Now, it is very, very interesting to notice, you see, the discovery of ZFN has a connection with, lower organisms. And here also, you have another system like tails, which are basically proteins which bind to DNA sequences in the host organism. To these tails, basically a nuclease domain, which is FOC1 endonuclease, which is also the enzyme which was used in the case of ZFNs, are engineered, and these tails or TALENs recognize and bind to a specific DNA sequence of interest. And when two TALENs are designed to bind to adjacent DNA sequences, the FOC1 nuclease domain will dimerize, creating a double-strand break at the target site. And we are already aware what happens following the occurrence of a double-strand break. The cell repair mechanism is activated, and then the events of insertion, deletion, and mutational events take place. In the lecture, we also have discussed about the applications of TALEN in the way genetic engineering and genome editing technologies in general are used, as in the case of, say, CRISPR-Cas9. Gene therapy is one of the most important area where TALENs hold significant promise. Other area is the functional genomics. Then we have its applications in agricultural biotechnology, and also TALEN is being used for model organism research, basically where genetically modified model organisms are created, such as mice and zebrafish for research purposes. These models help scientists understand fundamental biological processes. If you look into the various advantages of talents, we can, in brief, tell talents are having high specificity. It is also comparatively easy to design, and it has one advantage, which is remarkable: low off-target effects. We know that the earlier two genome editing technologies, ZFN is having more off-target problems. Talent and CRISPR having much lesser off-target problems. Talents comparatively exhibit a lower frequency of unintended genetic modification compared to the other two. However, there are certain challenges which we need to remember. The delivery of talents is very challenging. Efficient delivery of talents to target cells are challenging, particularly in clinical applications. There are size limitations. Talents are relatively large molecules which can limit their use in certain applications, especially when packaging them into delivery vectors. And there are other issues like ethical and regulatory issues. Talent also has raised ethical concerns and regulatory challenges, particularly when applied to human germline editing. So these are in brief the main genomic technologies that we discussed. So number one is the ZFN, then TALEN, and CRISPR-Cas9. So with this, I come to end of today's discussion, and over to CD. Or shall I continue to wait for people to come? Okay. How much time we have passed? Okay. Okay, let's wait. Hello, hello, CT. I don't know. Somebody will come, so I think we can stop, right? Yeah. Okay then. Thank you. Bye bye. Yeah.